So what we were looking at in that Punnett square just now are genotypes. So a genotype is the specific combination of alleles, so the specific versions of a gene that are carried in someone's genome. So if you were to sequence their DNA, sequence all their chromosomes, you'd find two versions, possibly two versions that are the same, possibly two versions that are different. Okay. The trait you actually observe, so like red hair, green eyes, being super tall, or like having blood type O, those are all phenotypes, things we can observe either with our eyes or, with a blood test, something like that. Presence of a specific protein would be a phenotype. Because remember that these genes are really just coding for proteins, right? The genes get transcribed onto RNA, get translated into proteins. And that's what causes these phenotypes. Okay, so when we're talking about genotypes, we're going to basically write down the alleles using letters. So when we have a dominant trait, we're going to mark that, and we're also going to mark which traits are recessive. So we're going to take the name of the trait. We'll see that we're going to use the color of pea flowers as one of our examples in a second. So sometimes pea flowers are purple. So our dominant purple color would be a capital P. Sometimes pea flowers are white, but that's a recessive trait. So we'd mark that recessive trait with a lowercase p. We're going to still use that letter P from the dominant trait so that we know we're tracking the same gene, because you might be tracking flower type, seed shape, seed color, and it's important to kind of know which genes go with which. Okay, so when we're talking about genotypes, we're also sometimes going to want to talk about whether they have two alleles or two versions of a gene that are the same, or whether they have two alleles, two versions of a gene that are different from each other. So when the two versions of a gene on those two homologous chromosomes are the same, we're going to call that person or that zygote, that cell, a homozygote. If we have two different versions, we're going to call that person a heterozygote. Further, we're going to specify when we're talking about homozygotes whether they have the dominant version, two copies of the dominant version, or two copies of the recessive. So if we had two capital Ps, two purple flower genes, purple is a dominant trait, so we'd call capital P homozygous dominant, or lowercase p would be homozygous recessive. So that would be a white flower. So remember, anytime the dominant allele is present, that's what you're going to see. So if you have a capital P, that flower is purple. But if you don't, this flower is white. So our heterozygotes are going to express the dominant phenotype. So those flowers will be purple, but they're going to have like a secret sneaky gene for the recessive phenotype. So in this case, white flowers. So we can infer, or like predict, or kind of solve the genetics of a person or an organism based on these phenotypes, based on these visible traits. Sometimes we're going to have to do a little more sleuthing than others. So if we have a homozygous recessive individual that shows the recessive trait, we know that they don't have any dominant alleles in there. So those genetics are going to be obvious to us. But if we see a dominant phenotype, that's not the case, and we'll have to do some more sleuthing. Okay. So one of the first people to sort of piece this stuff together, and actually piece this stuff together just by looking at phenotypes, they didn't have DNA sequencing techniques back then, was Gregor Mendel. He did a bunch of historic experiments with pea plants, which are really useful to think about genetics and think about the probability of genetics. And peas were really good for experiments for a number of reasons. One is that they grow and reproduce quickly, so you can do lots of experiments. Another is that they have a bunch of what we call dichotomous traits. which means they have a bunch of things where there are only two possibilities for a phenotype. So a flower in a pea plant is either purple or white. We don't have sort of an intermediate color. Now, if you think of a bunch of human traits, that's not always true, right? Like we think of kind of categories of hair color, right? Like brown, blonde, red, black. But there are all sorts of mishmashes in between. Not so for a dichotomous trait. The dichotomous trait just has two versions. So you see some examples here. Height in peas is dichotomous, either tall or dwarf. Seed color, seed shape. A lot of traits can be very easily categorized based on their phenotype as one thing or the other. Other thing that's super cool about peas is that you can breed a plant with itself. So like a flower on the right side of the plant can get crossed with a flower on the left side of the plant and make a new plant. And this is really important because it means that you can kind of control the genetics of the parent. So you can know that the two parents, because they're the same pea plant, have the same genetics. And that's useful for figuring out probabilities, for figuring out genetics. Okay, so the experiment Mendel did started with two parents that he knew were homozygous. Now he knew this based on basically flower shops, right? The flower shop guaranteed that every time you planted this pea, it was going to have purple flowers. All its offspring would have purple flowers, and no matter how many times you collected seeds and replanted them, you'd always see purple flowers. They'd also sell you a white version of the pea plant, right? So they'd promise you that every time you planted seeds from this pea plant, they'd always turn up white. If you bred that pea plant with itself, you'd still always have white flowers. Okay. So he started with two of these parents, which he called true breeding, but we know that this means that they were two homozygous parents. So the purple one was homozygous dominant, the white one was homozygous recessive. He named that generation the P generation. So basically they're the grandparents in this situation, but he thought of them as the parental generation. So that's what the P is about. Now, when he crossed these two pea plants together, so the purple one with the white one, he got what he called the F1 generation. So in the F1 generation, which he knew were hybrids containing one purple gene and one white gene from the two parents, he saw only one phenotype. They all had purple flowers. They all shared the dominant phenotype. Now, we now know that the reason for this is that dominant genes, dominant alleles, and in this case, purple, mask recessive alleles. So they hide them. So that's why we saw only purple individuals, because all of them had at least one purple gene. So from that purple homozygous dominant parent. Now, when he crossed the F1 generation with itself, so say this individual with this individual, you saw that he got a number of different phenotypes. So he got a lot of purple flowers, but he also got some white flowers. So this recessive gene showed back up in the F2 generation. Now we know that this is actually because each of those hybrids in the F1 generation was able to pass down either a purple gene or a white gene. So sometimes some of those plants in the F2 generation, just by probability, ended up with two white genes, which is why we had this recessive phenotype again. Now, every time he did this experiment, he saw this same ratio, actually, of 3:1 dominant to recessive. So this is actually why we use Punnett squares, or we can predict this result using Punnett squares. So here we see a Punnett square again. Instead of tracking chromosomes, I've put our trait into little pictures, right? So each of these hybrids in the F1 generation, we know is heterozygous, has a purple gene and a white gene. So if we split out, make our fake zygotes, say a zygote gets a purple gene and a purple gene from the F1 parents, and these zygotes in our boxes are the F2 generation, we could end up with a homozygous dominant individual. If they get a purple and a white from either parent, they're heterozygous. So that's 50% here of the offspring. Or if they get two white genes, here we see 25% of the offspring. So we see a consistent ratio in both genotype and phenotype. So every time we see purple in one of these boxes, we know that we have the dominant phenotype because dominant genes get expressed. So 3, 1, 2, 3. Out of our 4 boxes, 1, 2, 3, 4. 3 out of 4 show dominant phenotype. 3 out of 4 is the same thing as 75%, whereas 1/4 or 25% here are white, show the recessive phenotype. If we're tracking genotypes, we can see that 1/4 or 25% are homozygous dominant. 50%, 2 out of 4, or 1/2, if we reduce that, are heterozygous. Then 1/4 or 25% again are homozygous recessive. So as we do the problem set, we're going to do the same thing. We're going to track traits, but we're going to use letters instead of pictures. Okay. So from his experiment, Mendel came up with 3 laws of heredity, and he named them the law of segregation, the law of independent assortment, or the law of dominance. And they're also numbered. So first law, second law, third law. The first law is that every organism has 2 alleles of a trait, has 2 genes, 2 versions. Only one of these ends up in a gamete. And we know that's true because we know how meiosis works. That offspring inherit 1 allele or 1 version of each trait from each parent. I'm going to skip the law of independent assortment for a second, move to the law of dominance. So for the law of dominance, we know that recessive alleles get masked by dominant alleles. So an individual with a heterozygous genotype is always going to show the dominant phenotype. The law of independent assortment says basically that 2 traits can get passed down independently of each other. So if 2 traits are on different chromosomes, so hair color was on 1 chromosome, but height is on another chromosome, then the probability of you being short is not going to have anything to do with the probability of you being blonde. And that's true because chromosomes get passed down separately from each other. Genes on the same chromosome will get passed down together, but most genes are going to be passed down on different chromosomes from each other, so they'll be inherited separately. So as we're tracking genotypes and phenotypes, we have a couple of steps we're going to use to make our Punnett squares. First thing we're going to do is on the top and on the left of our square, we're going to write down the genotypes of the parents. So in this case, if we had a heterozygote, in this case, we're going to track height. So let's say T for tall. So capital T is going to be tall. Lowercase t is going to be short or dwarf. So here we see 2 heterozygotes, but you could have homozygotes as well. And you're going to write down the possible gametes that each parent could make, and you're going to put those kind of separate over 1 column or 1 row of boxes. So in this case, the capital or the lowercase. Our third step is that we're definitely going to draw this box with 4 squares. So here we can see our 2 separated letters. So this male would be a heterozygote, capital T, lowercase t. We've separated those 2 versions here because he could pass down either. Same thing for this female on the left. And then we're going to fill in the possible genotypes. So you could get a capital T and a capital T. Could get a capital T and a lowercase t. capital T and a lowercase t, or 2 lowercase t's. We're just going to do the same thing no matter what we have here for our genotypes. You have 2 capitals, you wouldn't end up with any lowercases here. Well, you would from here, from the female, but if you had 2 capital T's here, you'd get capital T from each parent as well. So you're just always going to figure out the genotype of the parents and then just do this. Okay. So then you're going to want to determine the relative proportions for both your genotypes and your phenotypes. So for genotypes, you're going to track down how many have capital T, how many have capital T, lowercase t, and how many have lowercase, lowercase. And you're going to convert that to percentages. So in this case, 2 heterozygotes, you end up with 25% homozygous dominant, 50% heterozygous, 25% homozygous recessive. That's what we've been tracking so far in this lecture. In your phenotype ratio, you'd have 75% have at least 1 copy of the dominant gene. So 75% have the dominant phenotype, 25% the recessive phenotype. So a thing we might want to do, if we don't know the exact genotype of an individual, we just know they have the dominant trait. So they might be homozygous dominant, but they might be heterozygous. We do what's called a test cross. So test cross is when we take an individual with the dominant phenotype, and in order to figure out their genetics, we cross that dominant phenotype individual with a homozygous recessive individual. So we always know the genetics of a homozygous recessive individual because we know that they have to have 2 recessive genes or we wouldn't see the recessive trait. So when we cross them with an individual with unknown genetics, but who has the dominant phenotype, if we see some offspring with the recessive trait, we know that our unknown individual must have been a heterozygote. So they must have had that recessive gene there somewhere, but hidden, or none of their children could show that recessive trait because they would all have a dominant copy of the allele if all that parent had to give was 2 dominant copies. So you could only end up with lowercase, lowercase if this unknown individual had a lowercase in the first place. So a test cross is going to tell us